Hi, my name is Anna Magos and this is my answer to question number 4. I will be providing the names of antimicrobial drugs and their effects. Antimicrobial drugs that inhibit cell wall. As we learned from chapter 3, bacterial cell walls are composed of peptidoglycan. Peptidoglycan is a macromolecule composed of polysaccharide chains such as the N-acetylglucosamine, NAG, and N-acetylmuramic acid, NAM, that are cross-linked by a short peptide chain between the NAM subunits. Beta-lactam rings are found in penicillin, and its function is to inhibit peptidoglycan formation by binding enzymes that cross-link the NAM subunits. This causes the bacterial cell walls to weaken, allowing water to move in the cell, causing it to lyse. And we can see the NAM and the NAG cross-linked by the bridge. And when the penicillin is taken, so it inhibits the formation of the ridges right here. Now, I'm going to talk about antimicrobial drugs that inhibit protein synthesis. Aminoglycosides such as streptomycin work by changing the shape of the 38 subunits, causing it to be misread. These drugs selectively target bacterial protein translation, and as we learned on chapter 7, ribosomes translate mRNA into proteins, and prokaryotic ribosomes are composed of 70S ribosomes with 30 and 50S subunits, as of eukaryotic ribosomes are composed of 80S ribosomes with 40S and 60S subunits. However, these drugs can be harmful to humans since their eukaryotic mitochondria contains 70S ribosomes, just like prokaryotic ribosomes. And we can see in the image that the incorrect amino acids are attaching to the ribosome, causing it to be misread. Now, I'm going to talk about antimicrobial drugs that disrupt the cell membrane. Polymers such as amphotericin B functions by forming a pore in the cell membrane. Lipid amphotericin B attaches to ergosterol, which is found in the fungal membrane. Humans can be susceptible to amphotericin B since ergosterol is similar to cholesterol. However, cholesterol does not bind as well to amphotericin B such as ergosterol. Most bacteria lack of sterol, but there are other antimicrobial agents such as polymyxin that also disrupts the bacterial cell membrane. And we can see that antiforicin B is attached to ergosterol, causing it a pore, and this causes the cell to lyse. Lastly, I'm going to talk about antimicrobial drugs that work by inhibiting the metabolic pathway. As we learned in chapter 5, metabolism is all the biochemical reactions that take place in an organism. Some organisms have different metabolic processes. For example, glycolysis is a unique chemical reaction for some organisms, and in this situation, antimicrobial agents are effective when there are different metabolic processes between a pathogen and a host. Sulfonamide cytostructural analogs from para-aminobenzoic acid, PABA, which are required for DNA and RNA synthesis. Sulfonamides compete with PABA and for the active site of the enzyme that produces dihydrofolic acid, causing a decrease in the production of tetrahydrofolic acid and thus DNA and RNA, which interrupts organisms' metabolism, causing the cell to die. Human metabolism is not affected by sulfonamides since they don't synthesize THF or from PABA. And we can see this is the metabolic pathway in which PABA is converted to tetrahydrofluoric acid, and this helps to DNA and RNA formation. Thank you.